genetically modified foods are?
Like tomatoes and things they do like to make them last longer when they ship them and stuff.
Right.
Genetic modification of food is a touchy subject at best. This is because most people do not understand how the process works, when in fact it is quite simple.
So let's say you grow tomatoes, and at night it gets so cold that by morning half your tomatoes are frozen, and now you can't sell them anymore. This is what scientists were able to change through genetic modification. They figured out a way to take DNA from one organism and put it inside a completely different organism to create a desired and specific outcome.
Scientists were able to use the genetic trait found in Arctic flounder that makes it possible for the fish to live in freezing water and transfer it into a new strand of tomato DNA, thus making freeze-proof tomatoes. Besides creating a more viable food product, the alteration of genes within food increases the amount of nutrients and vitamins found in the crops. This technique is known as nutritional enhancement. An example of this is genetically modified golden rice, which is derived from a white rice crop that has had a vitamin A gene from the daffodil plant inserted to address a vitamin A deficiency in developing countries. Advocates for genetically modified foods have also argued that modified crops are beneficial to the environment. This is because they are resistant to disease and are engineered to keep insects at bay. This means that farmers don't need to apply large amounts of pesticides and chemicals into their crops and the surrounding ecosystem.
Biotechnology is generally accepted as the use of living systems and organisms to develop or make useful products, or any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. In medicine, modern biotechnology finds promising applications in areas such as gene therapy.
Gene therapy is the use of DNA as a pharmaceutical agent to treat disease. The most common form of gene therapy involves using DNA that encodes a functional therapeutic gene in order to replace a mutated gene. Other forms involve directly correcting a mutation or using DNA that encodes a therapeutic protein drug rather than a natural human gene to provide treatment. Gene therapy was first conceptualized in 1972. The first FDA-approved gene therapy experiment in the United States occurred in 1990. Since then, over 1,700 clinical trials have been conducted using a number of techniques for gene therapy.
The idea with gene therapy for cystic fibrosis is that you would be able to introduce the kind of usual healthy version, if you like, of that gene into the cells that are affected, and that would produce the protein that's not working.
Although early clinical failures led many to dismiss gene therapy as overhyped, clinical successes in 2006 through 2011 have bolstered new optimism in the promise of gene therapy. These include successful treatment of patients with the retinal disease of leukemia and Parkinson's disease. These recent clinical successes have led to a renewed interest in gene therapy, with several articles in scientific and popular publications calling for continued investment in the field.
Modifying organisms for medical purposes has so far changed and saved the lives of millions, and it has done so from just about every possible angle. Genetic engineering has always been a controversial topic. It initially sounds very unnatural and is an occurrence that conflicts with Mother Nature. People worry that no matter what benefits might be introduced by engineering plants and animals or even humans, there's bound to be new problems introduced as well. What is it that makes this topic so controversial? Why do we categorize some genetic engineering acceptable as opposed to others? Organizations such as Greenpeace are strongly against the use of genetically modified organisms. They fear that the genetically modified organisms will integrate themselves with organic organisms, producing new forms and thereby permanently changing the environment. They also cite that there's too much unknown about the effects of genetic engineering to safely apply to our food sources. However, experiments in 1987 by the Committee of National Academy of Sciences have already concluded that genetically modified food pose no risk of damage to the environment. These experiments on tobacco and tomato were preceded by the engineering of bacteria to create insulin and interferon in the 1970s.